everybody. You're listening to Through Time and Clades. My name is Joan,
And I'm Albert.
and welcome back to Humanity of Prologue Season 2. In this episode, we're continuing our forward march through deep time to explore our branch of the Tree of Life, the Clade Hominini. This group includes the chimps and the hominins, the members of the lineage that includes human beings. And we'll be exploring the aspects of our physical evolution,
Hmm.
paying attention to traits that are acquired, which define our species, Homo sapiens. Discussions of cultural evolution, then, will concern most of the rest of this series. But before we jump in, how you been doing so lately?
I would say not bad. I've been very busy, but I've gotten quite a few things done, and I'm in a relatively productive mood at the moment, which is not always the case. So I'll take what I can get.
Oh, certainly, and likewise as well. Since we've been changing the format of how we work on Through Time and Clades, I've been investing more and more time into this lecture series of mine, and I got some pretty exciting things along the way that I'm excited to get to. Um, but alas, let's move on and see what we have to show here. So you may have seen images like the one on the left here that claim to show human evolution as a linear progress from chimps or chimp-like apes into anatomically modern humans. This iconography originally stems from an illustration by paleoartist Rudolf Zallinger, which he created for the 1965 volume Early Man for the Life Nature Library, called in the book The Road to Homo sapiens, but nowadays known as The March of Progress. The image itself is fascinating in its own right in a number of ways, like how the reconstructions of early apes and hominins have actually highlighted parts of their bodies to designate the known fossil material at the time. But the goal of the original work was not to showcase a literal step by step display of our evolution. Instead, Zallinger and the book's author, that is the paleoanthropologist F. Clark Howell, wished to show milestones of primate and human evolution in a temporal sequence, with all the species placed in upright positions to allow for comparative anatomy. But the general public had ended up looking beyond the artist's intent, nor bothered to read the original text, it seems. And now, even today, images inspired by this are perhaps the most visible symbols of evolution by natural selection in the world. And it's my hope that by the end of this episode, you will appreciate how complex human origins actually are. All right, let's move on to the next slide then. So, recapping from episode 2, we saw that the earliest apes, like the proconsolids, walked quadrupedally on their palms, much like the other monkeys. That is, they moved with chronograde locomotion, keeping the body in a horizontal position on the ground or in the trees. Over time, with the loss of the tail for balance and subsequent changes in the structure of the rib cage and upper limbs, apes switched to an orthograde locomotion, where the body is held vertically. This is especially seen in the ways that living apes climb and brachiate through the trees, using their arms to move from branch to branch, or even walking bipedally along the branches. As we look at the ape family tree, shown at the left, we see that a paraphyletic assemblage of fossil apes, known as Dryopithecines, seem to have been ancestral to the living great apes, or hominids. From there, the African apes, or hominines, branched off from the ancestors of orangutans, or pongines. And then later, the ancestors of gorillas split off, leaving us with the last common ancestor of chimps and hominins. So, when it is said that chimps are our closest living relatives, this is what is meant cladistics. Let's move on to the next slide. There's quite a lot that could be said about our relatives, the two members of the genus Pan. The chimpanzee is perhaps the most familiar of the two, living in a wide distribution across western and central Africa, north of the Congo River, while the bonobo, historically known as the pygmy chimpanzee for its more gracile body plan and somewhat smaller height, lives south of the Congo River. Now, as this episode concerns physical evolution, I'll be returning to these fascinating behavioral insights for both species at later times. But what do we know about chimp evolution? Well, at the moment, beyond genetic evidence, the only other source of information we have comes from fossils. And if you look at the bottom right at those two teeth, that is all we have. It seems unfortunate, but they do provide a key insight. So these teeth date to 545,000 years ago, within the Rift Valley of Kenya. Today, chimps aren't found this far east, and that tells us that their distribution was much wider in the past, and that they were living contemporaneously with our hominin relatives. We can only hope that more and better fossils are found of prehistoric chimps, but that still leaves us with the question of the nature of our common ancestor. So let's move to the next slide. Molecular evidence has waxed and waned over the years, but it seems that there was a period between 12.1 to as recent as 4 million years ago that members of both the chimp and hominin lineages were still seemingly interbreeding with each other, with some evidence adding the gorilla lineage to all of that mixing. And that has made pinpointing a date for that last split difficult. But many paleoanthropologists agree that hominins were well on their way to being a separate clade by 7.5 million years ago. The oldest known hominin fossils date to just a few 100,000 years after that date. But what about any older fossils than that? The record of Miocene apes is growing, but still contains a number of important blind spots, particularly when it comes to the possible locality of our common ancestor. Conventionally, based on our status as African apes, researchers have looked to the African fossil record for clues. But there's a huge gap between 14 and 10 million years ago, between the morphologically ancient proconsolids and afropithecids, and two controversial genera that have been proposed as members of the gorilla lineage. This has led some, like paleoanthropologist David Begun, to argue that we should be looking elsewhere for our common ancestor, like perhaps Western Eurasia. The fossil record for Miocene apes is much better at that time, as is seen in the graphic to the left, and with the very good fossil of a species known as Pyrolopithecus catalonicus to the right. This would suggest that climatic changes which affected the forest habitats of apes in the Miocene epoch pushed apes upwards into Europe, where it was less dry. And then once conditions improved, they re-expanded back into Africa and then gave rise to the ancestors of gorillas, chimps, and hominids. So let's move to the next slide from there. Uh, Regardless of where the common ancestor may be found, what can we say about what it looked like? Because it was assumed for many years that apes were, quote, more primitive than humans, well, researchers should look to gorillas and chimpanzees to find out about the earliest hominins then. Of course, we now understand and appreciate that these apes have co evolved with us for some 12 million years and that they would have undergone just as radical changes as we have. In one key example, we now recognize that the knuckle walking locomotion of gorillas and chimps are two separate adaptations to a more terrestrial based existence where retention of climbing behaviors was still important. Well, based on the most recent reviews of the Miocene ape and early hominin fossil record, the case is now growing that the immediate ancestors of hominins, and perhaps of other apes too, had adapted their orthograde postures to facilitate bipedalism. We can imagine them walking through the branches of the trees on their hind limbs, and when the need arose to move along the ground, then they could walk upright for at least short periods of time. This is known as facultative bipedalism, and it has been inferred from fossils of the earliest hominins like Ardipithecus ramidus. And it would mean that when the ancestors of hominins split from the ancestors of chimps, it wasn't so much that we stood up, but rather that we retained orthograde bipedal locomotion and just came to rely on it more than any other ape has nowadays. In one of the most recent surveys of ape evolution by Sergio L. Mejia and colleagues from 2021, the locomotory repertoire of a number of living apes was compared with data from fossil Miocene apes. And the team was able to hypothesize that our common ancestor, our last common ancestor, was likely moving in a way that was totally unlike humans or chimpanzees. It was capable of this occasional facultative bipedalism, but also moving around frequently on all fours. So now let's continue down the hominin lineage and meet the various members of our clade. Again, I must stress that this is not a step-by-step look at hominins as they evolve. It's not like as we move from genus and species to genus and species that they're going to all be evolving one into the other. No, this is rather a survey across different clades of hominins and what the fossils have to say about human evolution as a whole. I think I can safely say that the majority of these species that we're going to look at were probably not direct ancestors of Homo sapiens, but were more closely related species that were no less important. So let's move to the next slide and begin. The earliest known hominin is Sahelanthropus chidensis, which is represented from perhaps a few individuals that lived between 7.2 and 6.8 million years ago in Chad, north central Africa. Despite its importance as an early hominin, the analysis of this material has been rather closely guarded by its discovery team led by Michel Brunet over the 22 years since the first publication. As such, there's a lot we don't know about the species, and controversy reigns over whether it was bipedal or even whether it was a hominin at all, as features of the skull from bolt to base are by all measures more similar to chimpanzees and other non human apes than to known hominin. Now, phylogenetic studies to date have aligned it with hominins instead of gorillas, as some early work proposed, but the nature of bipedalism was primarily rested on the position of the fossils of the foramen magnum, or the hole in the base of the skull where the spinal column attached. Though, in the absence of other data, this is never a clear cut way to show bipedalism, it turns out,
Hmm.
as some living monkeys have been shown to have a so called bipedal foramen magnum on what is clearly a pronigrate body. In the last few years, partial ulnae and a femur have been reported and described, with the respective teams studying their remains not agreeing whether they show evidence of bipedal behaviors or not. At the very least, this alone suggests that Sahelanthropus still retained a possibly ancestral state of arboreal bipedalism with associated quadrupedalism. And of course, the study of the dentition shows that Sahelanthropus also retained the ancestral honing gap between the canine and premolar teeth, as seen in living apes today, and is indicated by wear patterns on the tooth enamel. But other details remain a mystery until the Brunet team returned with their seemingly secret analysis. And of course, rumor among the paleoanthropological community has it that there may be more specimens
Hmm.
currently unpublished. But until then, we're left with a few tantalizing clues about the biology of Sahelanthropus, which may have been as large as a living male chimpanzee. So let's move to the next slide. Next, we have a second genus of early hominin, Auroran, which to date contains two species. The first and the oldest is Auroran tuganensis, named after the Tugan Hills of Kenya, where it lived between 6 and 5.6 million years ago. A handful of remains were uncovered by a team led by Martin Pickford and Brigitte Sinew, and some of the bones reveal key details. Most notable is the femur, which shows the telltale morphology and angle of a bipedal hominin that was
Hmm.
moving more frequently on the ground on two legs, though the upper limb bones show a retention of arboreal locomotion. Also notable are the teeth, which show that the honing gap had been lost in this genus. The second species is one of the newest known hominins, Auroran praegens, also from Kenya, but dating to between 5 and 4.4 million years ago. This material had actually been known since the 80s, but was only recognized as a distinct member of this genus in 2022. And it differs from Tuganensis primarily in its dental morphology, but it does extend the lifespan of Auroran further forwards in time. Which, if we move to the next slide, we will meet another later hominin. So, contemporaneous with Auroran is the genus Ardipithecus of Ethiopia, again represented by two species. The earliest Ardipithecus cadaba from 6.3 to 5.2 million years ago is known from rather poor material, but the recovered teeth do show that this species had also retained the premolar canine honing condition. And this was lost in the younger and more completely known species Ardipithecus rhomidus of 4.8 to 4.4 million years ago. So it seems that the honing gap was lost independently among hominins if this is the case. But thanks to the specimen Ardi, which is shown here at the far right with a reconstructed skull, and the 15-year study from the team led by Tim White, this is the most well-known of the early hominins and the one that can give us the most information right now about how the first hominins differed from earlier apes. To start with, the rhomidus material includes the foot bones, with a large divergent hallux akin to what we see in most apes, and yet the sole and heel bones indicate that the foot was pushed off the ground as in how humans today walk. This is coupled with the state of the pelvis, different enough from the human condition, but with a shorter ilia than living apes. And the state of the limbs, nearly the same length but with long curved arms, shows that Ardipithecus has a mosaic anatomy. It could walk efficiently enough as a biped, but could also climb very well. And that this checks out with the older but less complete early hominins is a good sign that we may have the ancestral state for the first hominins, represented by the RD specimen.
Hmm.
So let's move to the next slide and see, where does that leave us for now? Our common ancestor with Ardipithecus and the other early hominins would have been at least a facultative biped that could move easily on the ground and perhaps in trees on two legs, though not exclusively. It would also have lost the premolar canine honing complex, as well as the reduction in the size of the canine teeth in both sexes. Now, this so-called feminization of the canines is a key distinguishing feature between living apes and humans, where in species like chimpanzees, males have proportionally larger and longer canines than females. Now, we do not have the time to go into the behavioral applications of this today. That's for another episode. But for now, we can see evidence for this all along the available early hominins. Thus, the earliest stages on the road from our common ancestor are a greater reliance on bipedalism and changes in dentition. Now, a curious side note to this story is that this ancestral state seems to have stuck around while the other hominins continued to dramatically change their physiology. Around 3.4 million years ago, the owner of the Bertele foot, shown here, roamed around Ethiopia with toe bones that show remarkable similarities to that of Ardi. Of course, there's nothing intrinsically special about this. Ancestral versus derived states are relative to the species being discussed, and there's no law in evolutionary biology that says that ancestral characters need to disappear as newer traits are acquired. Um, Before we move on, Albert, do you have anything you'd like to add?
Oh, not too much, but I've certainly heard about all the named specimens that you've discussed here, and you already start to get a sense of how rich our stem human fossil record actually is. Even though like some of these specimens are not the most complete in the world, we have quite a few of them, and enough to tell us quite a bit about their biology, which is certainly a lot more than what the chimps can say. So, yes, we will be seeing a very interesting picture as we move forward.
Oh, definitely. Let's move on then to the next slide. Much ink has been spilled on the interrelationships of the hominin species.
Hmm.
The standard practice of paleoanthropology has been using morphological comparison to parcel out similarities and distinctions between fossils to determine, their inclusion into a species or demarcate them as a new taxon. And this has led to a decades-long struggle to develop a taxonomic system that all researchers can agree with, the so-called lumpers versus splitters discourse, which extends into another similar struggle to make sense of this branch of our family tree. To date, there is little agreement among paleoanthropologists about just how our evolutionary map should be drawn, beside moving away from the long overthrown model that our origins proceeded stepwise with one species at a time. From there, several varieties of morphological analysis can be used in providing comparative studies that can then be applied to justify or discredit species designations and relationships. For example, a popular approach has been computer-based morphometrics, plotting measurements of anatomical characters from a number of specimens onto a data sheet to more visibly demonstrate the likes and dislikes and perhaps showcase patterns in the data. To the frequent comedy of others in the natural sciences, the use of phylogenetics or cladistics has been rather late to come in paleoanthropology.
Hmm.
As an approach, it can provide additional benefits to understanding hominin relationships by plotting character traits into their most likely relations and showcasing their evolutionary paths in a branching pattern, as seen in the approach showcased heavily in the last episode. At once, these methods can reveal key findings, as in the phylogeny to the right, which comes from a 2023 paper by Kerry Mongol and colleagues, which demonstrates, for example, that well-known taxa like Australopithecus are almost certainly paraphyletic, with respect to descendant taxa like Paranthropus or Homo. Now, this has been suspected for some time, however, and several researchers in the field, including Miv Leakey, Camilo Jose Calaconde, and colleagues, have made proposals to split this genus up. But so far across the board, most researchers have continued to use such older names. And there may perhaps be some understanding to this. The origin of species is a complex process and has been known to work under different conditions and scenarios, with several models proposed. For example, cladistics may imply cladogenesis, whereby populations split through varied means, and it's that which causes speciation, while anagenesis involves the gradual change in an interbreeding population from one species into another. There is evidence to suggest that while it's much rarer than cladogenesis, anagenesis can and has happened on islands, for example. And at that point, where would you put a marker saying that one genus begins and another ends? Does the naming of species then become, as many have argued, a subjective process of classification to make sense of data to human minds? As well, the increased use of ancient DNA has shown a deep history of hybridization that suggests hominin species boundaries may have been more fluid and open than once understood, and so has blurred this approach of distinct species delineation by suggesting that hominins underwent a mosaic evolution of lineages diverging, remaining distinct, and then coming together again to form new descendants. This is especially argued for the most recent evolution of our lineage, leading to the first Homo sapiens. And the question then becomes, how can paleoanthropologists detect such changes in the fossil record? In reality, this is something that is very hard to do and likely will not bear any definitive fruit at the moment without an increased sample of ancient DNA much further back in time than we have. Until then, I think the best course of action would be to use a variety of approaches on the widest possible data sets of specimens to come to a more objective understanding of just what we have, alongside the hunts, of course, for better and more complete remains. Albert, do you have any input onto this sort of discourse?
Yeah, somewhat. Yeah, I guess so. It's certainly something that those of us who study evolution through deep time have to contend with a lot. And as someone who doesn't study hominins or mammals of any kind, but studies phylogenetics and evolutionary history, it is a little baffling to me that anthropologists have been so reluctant to adopt the methods of quantitative phylogenetic analysis that most of us studying other types of animals have, and indeed having a robust phylogenetic tree really underpins a lot of the major research that we do in studying other animals or even other organisms in general. But it is good to see that is starting to change with several studies in the last decade or slightly more having come out trying to figure this out from a more quantitative perspective. So I definitely respect that. It is important to recognize the complexity of evolution and of biology in general in studying all kinds of topics in this field, and this is certainly a kind of classic illustration of that, really.
Oh, certainly. Let's move on then to the next slide. And of course, this will bring us now to the aforementioned members of the classic genus Australopithecus, sometimes understood as an evolutionary grade, which is shortened to Australopiths as a common name. In this area of our lineage, we find the first telltale signs of habitual or obligate bipedalism, of hominins that primarily walk on two legs, as seen in their hind limbs, feet, which is attested both skeletally and from trackways, shape of the pelvis, and the orientation of the skull on the vertebral column. The presence of ancestral anatomical characters in their curved fingers and lengthy arms and shoulder girdles seem to show that Australopiths still retained the ability to climb, forage, and perhaps rest in trees. They appear to have been ecologically flexible, inhabiting wooded environments as well as open grasslands across a wide swath of the African continent. And it's in this genus that we see the first members of homininae in southern Africa. They ate with an enlarged cheek teeth, with thick enamel inside heavily set mandibles. They may have even looked more like us than earlier hominins. So conventional restorations of Australopiths have made them look, have designed them to look right at home on the set of Planet of the Apes. But recent work on genetics of our parasites, like pubic lice, suggests that by at least 3.3 million years ago, the Australopiths on our ancestral line had significantly reduced much of their body fur. meaning that most of these species may have more resembled the restoration shown at the bottom right. But of course, I must stress that humans are not naked apes. We actually have the same number of hair follicles as the other apes. It's just that our pelage has reduced significantly and shortened
Mm.
to become, the hair on our arms and on our bodies and on our legs. Um, and of course, that can vary too between populations. But as a whole, we don't have fur. That's really the big difference here. But we're still very hairy. Now, behaviorally, though again we'll explore this deeper in a later episode, Australopiths also seem to have given far more attention to their young than any other apes before them or since. Strangely, analysis of tooth crowns and the few remains of young that we have show a pattern of fast growth similar to living chimpanzees, but a much slower trajectory of brain development, closer to what we see in humans today. And that would imply that baby Australopiths were far more vulnerable to threats and needed a longer period of time to learn skills and other appropriate behaviors, something that their mothers and perhaps their families and communities could give them. And again, reiterating by the conventions of cladistics, the genus Australopithecus is technically paraphyletic. But I'm going to break with the approach that we used in the last season of Humanity: A Prologue, and I'm going to retain the use of Australopithecus in describing most of the members here. That is until changing name conventions become more commonplace and accepted in the field. So let's move to the next slide and begin our survey of them. The oldest known Australopith is the 4.3 to 3.8 million year old Australopithecus annamensis from Ethiopia and Kenya. Initially known from rather fragmentary remains, recent work by Johannes Haile Selassie and colleagues has finally given a face to this species, revealing a form very much similar to later species, save for distinctions in tooth morphology. As such, much controversy rests on whether Annamensis represents a distinct taxon or whether it was an earlier ancestral trait. For the more familiar species on the next slide, Australopithecus afarensis, currently dated to between 3.9 and 3 million years ago.
Mm.
The famous Lucy specimen, known as Dikenesh in her native Ethiopia, and shown here third from the right, belonged to the species. But in contrast to her status by both the general public and the young Earth creationists, she was far from the only known individual of this species. Currently, there's something like 300 individuals that have been described since the 1970s, including adults and juveniles, both male and female. Included in this collection is perhaps the controversially named specimen described as Australopithecus baral-ghazali from Chad, which would then extend the species range into the center of the continent. What we have of afarensis shows hominins that, at adult height, stood between 1.6 and 1.2 meters tall. It's been argued in the past that the apparent differences in canine and body size between the presumed male and presumed female afarensis related to a strong sexual dimorphism akin to living gorillas. But some work by Philip Reno and Owen Lovejoy in 2015 on our sample of remains has failed to support that hypothesis,
Mm.
pointing out that afarensis sexual dimorphism actually lays within the modern human range, which exists but is markedly small compared to the other apes. As well, other features of their anatomy are quite human like. In a rare example of homoplasy or convergent evolution for hominins, Australopithecus afarensis had developed limb joints more comparable to the later members of Homo than to other Australopiths, which gave them a greater control of movement in terrestrial settings, something that muscular reconstructions also support. Still, retention of traits linked with arboreal behaviors are still present. The scapula of afarensis has been described as gorilla-like, and the length of her toe bones would have allowed for an easy grip on tree branches. So moving on to the next slide, we have two other little-known species of
Mm.
East African Australopiths. The first is Australopithecus garhi from Ethiopia 2.5 million years ago. This species is distinguished primarily by the size and structure of its teeth, no surprise there, which are larger and resemble those of Paranthropus, hinting at a close relation to that clade. Unfortunately, all we have that is for sure attributable to Garhi is the fragmentary skull, shown in a reconstruction here to the left. But there were other specimens found nearby that could be linked, but definitely not at this time. One of those include a femur, which has a morphology indicating hind limb proportions more closely akin to genus Homo than the earlier Afarensis, which would fall in line with recent phylogenetic work, placing it closer to that line than to Lucy's species. The second is Australopithecus diarrhea, also from Ethiopia, but dated to 3.5 to 3.3 million years ago. This is one of the newest recognized australopiths and has been marred in controversy as to
Hmm.
whether it should be realized as a distinct form uh, or whether it belongs to Afarensis or not. The original description by Johannes Halselassie and colleagues pointed to the preserved maxilla and mandibles, which protruded forwards in a way not seen in Afarensis fossils, and with smaller teeth to boot. In fact, very recent work from 2024 by Hester Hedengraf and colleagues seems to have added further support for its distinction, using morphometrics to show that the original traits used in the description of the species were indeed different.
Hmm.
So let's move to the next slide then. So far, our hominin journey has remained in roughly the upper half of Africa, but now moving southward, we also come across the earliest discovered Australopith, Africanus. This species was originally described by Raymond Dart in 1925 from the tongue child skull, shown at the top left, but is now known from several good specimens across South Africa, dating from 2.3 to perhaps as much as 3.7 million years ago, if recent dating approaches at the historic Fontaine caves are validated. In many ways, Australopithecus africanus is more human-like than other australopiths, especially in its postcranial remains. And phylogenetic work shows it actually sharing a common ancestor with both Homo and Paranthropus. However, Africanus is also unique in many respects, having limb proportions more like living apes, a further display of the mosaic anatomy characteristic of Australopiths. Recent research on the dexterity of the fingers also strongly supports the possibility that Africanus, and perhaps other Australopiths too, have the ability to maneuver their digits in such a way as to craft and hold stone tools, which is behavior we'll explore at a later date. Moving on to the next slide from here. In the late 1940s, Raymond Dart had recognized a contemporaneous species to Africanus, which he called Australopithecus prometheus, today encompassing a collection of specimens from caves in South Africa dating to between 3.67 and 2.6 million years ago. And that includes the recently extracted specimen STW 573, or Littlefoot, which is shown here on this slide, which is without a doubt one of the best and most complete Australopiths currently known. This is a controversial species, and many researchers agree that the material falls within a wider variation of Africanus. But the team led by Ronald Clark, who had recovered the Littlefoot material to begin with, have strongly argued that the overall larger dimensions of the skeleton compared to Africanus justify its distinction. Needless to say, the name is not without taxonomic concerns, but regardless, the Littlefoot specimen is absolutely beautiful and has been revealing many exciting secrets of Australopith anatomy. For example, the pectoral girdle shows a similarity to living apes that climb trees, just like in Afarensis, with the caveat that the movement of the arm worked best in arboreal settings with the hand above the head. Um, in another example, the os coxae, or the hip bones, shown here at the bottom right, actually reflect a human-like morphology akin to obligate bipedalism. So moving on to the next slide from here, we have the case of the 1.98 million year old Australopithecus sediba
Hmm.
from Malapa Cave in South Africa, which is a fascinating one. It's only known from two specimens, and the team led by Lee Berger have described the remains as very similar to Africanus in size and dimensions, but as well something unique on the South African landscape. Analysis of the lower limb bones points to Sediba moving with a very different form of bipedalism compared to other australopiths, with the legs being held in a hyperpronated state, with the ankles pointed at inward angles, and the overall shape of the pelvis more resembles members of early Homo than other australopiths. In fact, while there are clear indications of some form of evolutionary relationship between Australopithecus africanus and Sediba, many phylogenetic studies have placed it closer to the genus Homo, and sometimes within it itself, which would make it Homo sediba. Though those specific results have been contested by the fact that the skull material used for phylogenies, which is the most complete out of the two, comes from a juvenile specimen that might reflect ontogenetic changes.
Hmm.
Basically, if we had more complete adult skulls, the results might differ somewhat. Now, judging from previous work across all the known species of Australopithecus known so far, a recent review paper by Zaracine-Almenzeged from 2023 presents a possible scenario for the evolution of the genus Australopithecus. It's suggesting that most species ultimately evolved from the widespread afarensis through a combination of anagenesis and budding speciation across a period of time from 3.7 to 2.9 million years ago. The latter date, of course, would be when it gave rise to Paranthropus and Homo. Um, Interesting idea. Of course, it'd be nice to see more phylogenies test that result. But there is another species to consider before moving on to those two genera. If we move to the next slide, um, that is being, of course, Kenyanthropus platyops from Kenya, 3.5 to 3 million years ago. It is mainly known from a crushed skull that, nonetheless, is so uniformly crushed that the overall shape is probably not too far off from reality. Right away, the uniqueness of Kenyanthropus is apparent, though not exaggerated. Compared to other Australopiths, while the face is not fully prognathic but rather flattened, it does fall somewhat within the variation known for Australopith skulls, and it has been suggested to be subsumed into Australopithecus to reflect that. In the interest of cladistics, however, I'm personally fine with keeping this genus distinct. Otherwise, there is no known postcrania to go by. So let's move to the next slide from here. The genus Paranthropus seems, again, to share a common ancestry with Homo, and so it is only related to our ancestral line. Nonetheless, the so-called side branch represents a fascinating example of unique speciation among hominids. It survived the longest out of all the Australopiths, dating to between 2.9 million years ago to 800,000 years ago, and having ranged across eastern and southern Africa as four species: Ethiopicus, boisei, robustus, and the very recently described capensis. And goodness gracious, the paper that described that came out like 2 days before we started recording, so the timing could not have been more perfect. Um, they are well distinguished from other Australopiths, primarily in their skull morphology, which is massive. Large cheek teeth, wide faces, and prominent sagittal crests on the top of the head for supporting huge chewing muscles. Traditionally seen as nutcracker men who subsisted on tough and nutty foods compared to their more omnivorous cousins, nowadays researchers who have studied Paranthropus with newer technologies have shown these hominins as having more varied diets, with some species like boisei including more gritty C4 plants like grasses, and perhaps grazing for hours at a time. Otherwise, their overall size was on par with other Australopiths, and they probably looked very much like them too, as well as relying on the same repertoire of bipedal and arboreal behaviors, something that's supported by studies of the available postcrania. Before we move on, do you have anything you'd like to add about all the Australopiths that we've covered today?
Certainly quite a group of superstar STEM humans here. Of course, there are a few like relatively obscure species too, but yeah, certainly here we're seeing quite a number of species or even specific specimens that just about anyone who has even a passing interest in human evolution is likely to have heard about. And it is interesting to hear about like all their differences and recent efforts to clarify their evolution and relationships to each other, because it does raise a question that you brought up both earlier in this episode and in the last series of how to apply names to these different taxa, right? It's like you kind of mentioned there, calling all those different species Australopithecus. It kind of obscures the fact that they're not all the same, and indeed some of them might be more closely related to us than to other so-called Australopithecus. Like the example of Sediba is really interesting, in particular with the discussion going on there. In fact, I think in the previous season of the series, you actually went with calling it Homo Sediba, if I recall correctly. Of course, as you mentioned, that's far from a settled thing, but it does go to show like the vast amount of diversity we are working with here. Yeah, no, this is a really interesting part of the human family tree. I think certainly they do deserve the attention that they get.
Oh, absolutely. And you're absolutely right. Yeah, the naming discourse is of great interest to me, obviously, because one of the things that I believe strongly about biology is calling things by the right names. But obviously, given the unequal distribution of quality material between all these species, it's very hard to get sort of the rigorous anatomical approaches that we can use for other fossil groups. Although rest assured, like there are researchers, as I mentioned before, who have been working on these specimens and have already proposed names that could be available for use if the majority of the field has decided to go along with abandoning Australopithecus as a wide encompassing name. Which again, yeah, you're right. In the last season, I did do this in a sense, which is why we called Australopithecus afarensis, for example, Prayanthropus
Mm.
afarensis, because that is an available name. Um, for those curious, should that come to pass, then only Australopithecus africanus and any related taxa to that in its clade would keep that name, and everybody else would require a different name. But I think regardless of that, one of the most fascinating aspects of the Australopiths is how many species lived at the same time
Mm.
and in the same environments. Again, kind of going against this idea of a stepwise evolution with one species turning into another, we find many sites in East and Southern Africa where multiple Australopiths were sharing the landscape. And, you got to wonder, how are they interacting with each other? Were they interacting with each other? Um, how did their behaviors translate to their interactions or lack thereof? These are all great questions and certainly ones I'd like to explore, see more research on as the field continues on. But alas, we should move on from here. Let's move to the next slide, and we can finally arrive at the genus Homo, which
Mm.
is home of one of the biggest controversies at the moment regarding its process of origin as a genus, which I'm going to take here, of course, to mean humans proper. We know from the previous analysis of morphological traits in earlier Australopiths that by the time the earliest ancestors of Homo arrived on the scene, they had acquired obligate bipedalism, supported by feet with inline toes, as confirmed by both fossils and tracked ways. They also had more elongated thumbs, which gave even better dexterity and precision grip to the fingers. We also find within the skull some key differences, notably that the cheek teeth have become slightly smaller in size, though a recent analysis from 2024 indicates that early Homo retained strong similarities in form to the Australopiths in their enamel and dentin than has been previously understood. It's also with our members of the genus Homo that we start to find evidence in the fossil record of what is called M3 agenesis, which is when the wisdom teeth do not form, though the ultimate origins for such a trait in certain individuals is still unclear. Much has been speculated about the skull and brain, but it is apparent that there was some reorganization in both tissues and proportions, with others remaining ape or Australopith like, particularly in the frontal lobes. It should be stated, not incidentally, that early research that you may have heard about suggesting a loss of function in the myosin gene, MYH16, which had been dated to around 2.4 million years ago, led to a reduction in chewing muscle mass and dental size, and in parallel aided in the expansion of the brain. That has now more or less been discredited.
Mm.
And I'll link to an article about this in the references doc here. Um, for the skull as a whole, though, it does show an allometric scaling in the vault size, which can also be detected when looking at hominin skulls from above. So, one way that I learned in my biological anthropology class to tell skulls of Homo apart from other Australopiths is when you look at them from above, you can see a characteristic pinching between the brain case and the brow that is seen in Australopith skulls. You don't find that in genus Homo. It's a much smoother and less apparent transition from those parts of the cranium, which is kind of neat. Somewhat more uncertain is a possibility that the white sclera of our eyes became a fixed condition by the time of early Homo. So we're alone among the living apes in having eye whites, which experiments have shown play a role in nonverbal cues and signals between individuals, helping others focus on items in the direction of another's gaze. In contrast, the other apes have a diverse range of scleral colors, on occasion white, but often black or brown, which makes gaze recognition more difficult. This shift implies that the ubiquity of white sclera in humans reflects a shift towards greater communication and cooperation at some point in the distant past. And as I mentioned before, the shift to more altricial young in Australopiths may indicate the start to this change. So far, the earliest member of the genus Homo has been identified as the 2.8 to 2.75 million year old mandible from Ledi Geraru in Ethiopia. It has not been assigned to any known species at this time, but the authors of its description, Brian Villamore and colleagues, note substantial similarities to known early Homo jaws and teeth than to Australopiths, though they are also not too distinct, showcasing a clear transitional sequence, if you like. By 2.35 million years ago, as shown on the following slides, though, our fossil record for Homo becomes much better and more complete. So let's move to the next slide and meet some of these guys. The earliest known species in the genus Homo are Homo habilis, or Homo habilis, from eastern and southern Africa, 2.35 to 1.65 million years ago, and Homo rudolfensis from Kenya, 2 to 1.95 million years ago, which in recent years has been more and more understood to be distinct from Homo habilis. Rudolfensis is currently known definitively from its large skull and some mandible material, which is noticeably flattened compared to both Australopiths and habilis, and has a different shape to the dental arcade or the sweeping curve of the jaws than other species. Now, there is some material that has been found nearby that may be associated with the skull, including a humerus, femur, and pelvis, though the features of these remains show a connection to Homo erectus, too. Homo habilis is known from more material, including postcrania, which demonstrates that the overall body form and proportions of early Homo had remained remarkably similar to the Australopithecus, showing that species like habilis were still capable of arboreal behaviors. The OH8 specimen, a partial foot, shows evidence of the characteristic ring in the steps of living humans that allows for a greater endurance during more quick-paced movements. The accumulation of ancestral traits in these species compared to later forms like Homo erectus has led to a lengthy discourse about whether the allocation of these specimens into Homo is justified, or whether they should be recognized as perhaps late Australopiths or even their own genera. As well, discrepancies in the material assigned to Homo habilis itself, now and in the past, has also led to questions about whether they represent a single species or not. So, a big morphological analysis by Ian Tattersall and Jeffrey Schwartz, as well as recent work by Fred Spoor and colleagues, indicates a so-called deep-rooted species diversity for early Homo, and a recognition of perhaps four or more distinct varieties within the sample of early African Homo, to the exclusion of Erectus, with anthropologist Darren Curnow having argued controversially in 2010 that the South African material is best understood as a separate species called Homo gautengensis. Of course, it is very well possible that the origin of early Homo was more complex than expected. And indeed, when we look at one of the most familiar hominin species over in the next slide, well, we can find that is more certain. So, taking lessons from the massive 2023 review article by Susan Anton and Emily Middleton, for this series, a number of material in Africa and Western Eurasia, and Eastern Eurasia as well, will be recognized as belonging to one diverse and long lived species with numerous genes exhibiting polytypic morphology that we're going to call Homo erectus. It is in members of this species from 1.77 million years ago and onwards that the expansion of the brain really begins to take effect, with the earliest members dated to southern Africa from 2.04 million years ago. African Homo erectus ranges until about 1.4 million years ago and encompasses a number of diverse specimens, including the famous Nerkatome or Turkana boy, a juvenile that was about 8 years old when he died. It's from material like this that has somewhat misled researchers into thinking that Homo erectus was far more like living humans anatomically than originally thought. Essentially, they would have been modern humans from the neck down. But Recent studies have reassessed the total material for Homo erectus, particularly in its earliest fossils like that from Gona, Ethiopia, dating to 1.55 to 1.26 million years ago, and our forthcoming Georgian material, and have shown that rather than being a step above Homo habilis in derived features, Homo erectus should be better seen as one among the representatives of early Homo, with overlap in forms and even in time with habilis. In fact, we have some fossil sites that show that Homo erectus, Australopiths, and Paranthropus were all coexisting within the same environment. The species' stature and limbs were relatively short, though there was clear anatomical evidence for a more refined bipedal locomotion. And footprints attributed to this species show weight distribution and morphology very much like living humans. Examination of the rib cage also shows a more Australopith-like arrangement than that of more recent humans. On a related note, the recognition of all the African material as Homo ergaster does not seem to align well with observations of morphological overlap with material in Eastern Eurasia, as well as similar research hinting at, again, a greater diversity among the African material than should probably be recognized as one species within Homo erectus. It seems that we have quite a confusing situation with early Homo. So let's move on to the next slide from here. We have the famous Dmanisi hominins, again included in Homo erectus here, but sometimes separated in their own species, Homo georgicus. These remains date to between 1.85 and 1.77 million years ago, and along with a very recently described material from Romania, 1.95 million years ago, indicates that early Homo had a presence outside the African continent very early on. That has been described as, quote, widespread, though perhaps intermittent. The Dmanisi material represents 5 individuals, including an elderly specimen that had lost the majority of its teeth, but did not succumb to starvation, which has implications that we'll explore in a later episode. A plethora of stone tool remains and butchery sites in France, Italy, the UK, and the Netherlands, dating to 1.8 to 1.7 million years ago, may perhaps belong to Homo erectus as well, but we just lack the definitive material fossils to test that hypothesis. So let's move to the next slide from here. At last, we come to the most familiar specimens of Homo erectus, those of the so-called Java and Peking, or should we say Beijing men. These are early 20th century names associated with the discoveries of Eugene Dubois in Java, Indonesia, in 1891 to 92, and with Davidson Black and Otto Karl Josef Zdansky in Jakutian, China, in 1927, which nowadays are just the tip of the iceberg in terms of Homo erectus material across eastern and southeastern Eurasia. As far as the taxonomy is concerned, these specimens would hold the most authority to be known as Homo erectus, what we call Homo erectus sensu stricto. And so, if a similar situation occurs with these specimens as with Australopiths that we mentioned before, then this material would keep the name Homo erectus, and all the others would need to get named. Looking at the totality of the evidence available nowadays, it seems clear that Homo erectus is among the earliest hominins to have reached the easternmost edge of Eurasia, with the oldest known remains being the Mojokerto and Sangiran specimens of Indonesia dating to 1.81 to 1.6 million years ago. They show clear anatomical features associated with the African and Georgian material. But curiously, there are also distinctions with the Eastern Eurasian material in China that has led some researchers to suggest that perhaps there were multiple dispersals into this part of the world. One that established the earlier Southeast Asian material, like Sangiran, and another that gave rise to the Chinese populations, like the Beijing man, that perhaps is related to material from Java, like the Yandong Solo man, which represent the youngest material from this species, at between 117 and 108,000 years ago. Of course, there is a noticeable gap in the fossil record between Indonesia and China that may confirm, invalidate, or further complicate this model, which of course rests on assumptions that Africa and not, say, Southwest Asia could be the hub of dispersal for the species. What can be more certain pertaining to this model is that some Chinese specimens, like Guanlongbing from Lanshan, dated to 1.63 million years ago, show dental morphology and cranial capacity that links it with both the Georgian material and the later East Asian remains. And as the earliest Homo erectus fossils in China, they could represent what those ancestral populations may have looked like. Now, like I said, the fossil material for Eastern Eurasian Homo erectus is vast, and it includes a number of fascinating glimpses. Notable examples include the aforementioned solo men from Nyangdong in Java, which have such a derived anatomy from earlier Javan material that some past researchers had thought that they were perhaps ancestral to modern humans in the region, which is an idea more relevant for discussion in the next episode.
Hmm.
But in recent years, there has been some renewed discussion about whether the solo specimens should be rendered as a distinct species that had evolved from Homo erectus, which would then be known as Homo soloensis. Of course, that lacks a consensus at the moment. It is also notable that most other Chinese material from this species have a so-called pinched-in posterior to the skull that is not shared by other remains, while much of the Java material shows a notable increase in cranial capacity over time. So it's clear that taken as a whole, Homo erectus was a very successful species of hominin that expanded its population right across Afro-Eurasia to such an extent that different populations seem to have begun the process of speciation. In regards to this species described in the next slide, that speciation may have been taken to the extreme. So let's move on from there. I'm referring, of course, to the famous hobbit of Flores, Indonesia, Homo floresiensis. Fossils from this relatively new species include a number of individuals, including the flow specimen shown here, that date to between 190 and 50,000 years, so very recent in time. Much of that material hails from the sites of Madamenge and Boa Lessa, but archaeological materials from Wolosenge hit that potential ancestors may date to 1.2 million years ago, and we now have additional fossils from Madamenge from 700,000 years ago. The most prominent feature of Homo floresiensis is that they were tiny for a member of Homo, with an estimated adult height of only 1.1 meters. That's 3 feet 7 inches tall. For comparison, people with dwarfism are taller on average by several inches, and ethnic groups like the Mbuti of Central Africa are taller still. A better height comparison for this species would be female Australopiths like the Lucy specimen, and that's not the only link proposed by some researchers. Homo floresiensis was linked with our genus in the original 2004 description by Peter Brown and colleagues because features of the skull and teeth were most similar to members of our genus and to Homo erectus in particular, especially considering the low-built cranium and the angled or flexed base of the skull. If this connection is correct, then what we would have is a case of island dwarfism, with a population of Homo erectus becoming isolated on Flores and gradually shrinking in dimensions to accommodate efficient resource use on such a small location. However, other researchers point to the post-cranial, especially the limbs and the feet, which are rather long and more like those of Australopiths than they are to the genus Homo. This is in contrast to observations that the jaws do not show the condition for large cheek teeth in Australopiths, nor does the skull have the classic prognathism either. In fact, to yet other researchers, what we really have here is a member of early Homo that is distinct from Homo erectus and perhaps evolved from that before that species diverged. Maybe a better link would be placed with Homo habilis, for example. That would make the small stature not as exaggerated as to be explained by island dwarfism.
Hmm.
So the most recent paper to dig into all of these weeds of discourse comes to us from Yokusei Taifu and colleagues from 2024. And looking at the complete remains from across the Flores sites, they found that the evidence best showcases a link with the earliest Homo erectus in Java, like Sangiri. Now, this will no doubt be far from the last word on the subject, but it does re-emphasize the morphological diversity of early Homo, including Homo erectus, that could explain why a hominin like this looks like such a mosaic. Now, the second species described here is Homo luzonensis, only described in 2019 from Calao Cave on Luzon in the Philippines, hence the name. They had initially been dated to as young as 67,000 years ago, but recent redating has made them older at 134,000 years ago instead. While the available material for this hominin is scrappy, some teeth, foot bones, it was enough for the research team of Florent Detroit and colleagues to distinguish it as a new species. It was another dwarf of a hominin with cheek teeth that were smaller than even Homo floresiensis, though how small in height we cannot determine with so few fossils at this time. Likewise, what little phylogenetic relations we could detect are difficult to pin down beyond its naming as a member of Homo. True similarities do provide a possible link with Homo erectus, so maybe this is another island dwarfism event. Of course, this is all brand new territory, and until more fossil excavations are done for the early Pleistocene of Eurasia, much of the hominin family tree here will remain with tentative but unsolid links. So let's move to the next slide and let's look at Homo naledi. Homo naledi has become quite the newsmaker in the last few years, given the massive amount of work done by Lee Berger and colleagues since they discovered and named the species in 2013 from fossils painstakingly recovered from the Rising Star Cave in South Africa. The current plethora of remains dates to between 335 and 236,000 years ago, which make them contemporaries with the earliest Homo sapiens and its ancestors. Homo naledi has been described as small-brained, having a cranial capacity of only 465 to 610 cubic centimeters, making them outliers among the hominins of the time. For comparison, Homo floresiensis' brain was only 380 cubic centimeters. And so it's more comparable to something like Homo habilis or even Georgian Homo erectus. However, analysis of the endocast, so the actual shape of the brain that you can get from the skull, points to a hominin with a forebrain that was more similar to our own than to members of Homo erectus, including an expanded Broca's area, which is linked to planning responses to speech. Likewise, the enamel on the teeth and the eruption of the second molar tooth shows more similarities with living humans than any other hominid. The brow and parts of the cranium bear resemblances to Homo erectus, while the forelimbs and shoulders exhibit features seen in Australopiths and perhaps conferred similar abilities for greater arboreality. The hind limbs are those of an obligate biped, however, with the trademark characteristics of strong foot support and a capability of long distance locomotion. So, all in all, this is a very strange hominin, which little in the way of phylogenetic analysis has been able to place it anywhere safely on the Homo clade. It's been allied with more derived Homo to the exclusion of Homo erectus, or it's been shown to be nestled among more early Homo generally. There is one potential fossil that may hold a key link, which are some skull fragments from Olorgo-Salale, Kenya, dated to 900,000 years ago. A 2020 reconstruction analysis pointed out the most similarities to this material to Georgian Homo erectus and Homo naledi, which raises the possibility of these fossils representing perhaps an early member of the lineage that's ancestral to the rising star fossils. But either way, the sheer volume of fossil material for this species makes Homo naledi one of the best sampled extinct hominins currently known. Before we move on, is there anything you would like to add about early Homo?
Yeah, quite a varied bunch for sure. Certainly been very exciting to see many of these finds come out in recent years. I certainly remember reading news articles about Homo floresiensis, and of course Homo naledi was a big deal too. I look forward to seeing even more work being done on all of these.
Yeah, absolutely. Me as well. What was really fun about Homo naledi was, yeah, I remember when it was announced too. In fact, I was in human evolution course at the time, and Lee Berger's team had made the excavation available to watch on live stream. So actually, for a brief moment in one of the classes, we could actually watch the researchers at work pulling out all these small bones and commenting to the live stream, which that's such a great example of science communication. We'll talk a lot more about Homo naledi as the series goes on, because I'm sure there are a lot of questions about its behavior that have come to light in recent years that are quite controversial, to say the least. That's not the place for that. Let's move on to the next slide. Um, Now, at last, we come to a point in time when the direct ancestors of Homo sapiens could be found. It is arguable that the wide ranging Homo sapiens gave rise to our last common ancestor with other hominins somewhere in Africa or Eurasia, but our information going forward in time becomes, no less messy. There is a plethora of fossil material from the Middle Pleistocene or the Shibanian age that has proven difficult to sort out by paleoanthropologists wishing to establish evolutionary links between specimens. This so-called muddle in the middle, which is a term coined by Chris Stringer, has seen increased attention in the last few years, including since we recorded season 1. And it looks like we may finally be getting somewhere, slowly. At any case, our last common ancestor with other hominins, like the Neanderthals and the Denisovans, likely lived somewhere within the last million years or so, but no younger than 430,000 years ago. Before we explore some of this material, it will be good to once again see where our ancestral characteristics lie at this stage. Hominins had by now evolved a cranial capacity exceeding 1,000 cubic centimeters, sporting large skulls with big brains. Our ancestors' bodies had by now acquired a somewhat more modern configuration than, say, Homo erectus, with a lengthening of the lower limbs that allowed for even better locomotion. Curiously, we now have brand new evidence from 2022 that our infamous fusion of chromosome number 2, located in our genome, may have happened right about this time, at an estimated 900,000 years ago. Of course, it is this fusion of chromosome number 2 that makes humans distinct today from the other apes, where they have that chromosome split into two. One species, which paleoanthropologists have somewhat more confidence about, that predates the muddle in the middle, is Homo antecessor
Mm.
from Spain, 1.2 million to 770,000 years ago. Sometimes referred to as the boy of Grandolina, although recent work nowadays suggests that we may be looking at a girl of Grandolina instead. The sequencing of its dental proteome, or the signature of all the proteins an organism's cells can make, has allowed it to be placed into a tentative phylogeny that shows this species diverged before the common ancestor between Neanderthals and Homo sapiens, though it must be remembered that the skull is from a juvenile individual. It has been noted that Homo antecessor had a remarkably modern human-like face than that of, say, a Neanderthal, which has led to hypotheses that either the facial proportions of living humans represents an ancestral character state that the Neanderthal lineage steered away from, or that living human-like facial anatomy may have convergently evolved in Homo antecessor. These are all good questions. Let's move on to the next slide, though. Our survey of the muddle in the middle must necessarily start in Africa first, because there has been strong evidence to suggest that the ancestors of Homo sapiens evolved there prior to 300,000 years ago. Our earliest hints to human evolution beyond Homo erectus are represented by the cranial fragments of Gombore II from Ethiopia, dated to around 850,000 years ago. These were described in 2016, and these fossils are the first to show a clear transitional anatomy between African Homo erectus and later hominins when compared morphometrically, in particular to the Broken Hill skull from Kabwe, Zambia, with its enormous cranium and brow ridges. These remains have recently been redated 299,000 years ago, which is much younger than was initially proposed. With this date in mind and a re examination of the morphology, it doesn't seem likely anymore that the Broken Hill individual was part of a lineage ancestral to Homo sapiens. The Undutu skull from Tanzania is older, around 450,000 years ago, and was once also thought to relate closely to Homo sapiens, but a very recent reconstruction has revealed that the original restorations had actually exaggerated those sapiens traits.
Mm.
Now, while a phylogenetic relationship could not reasonably be established at present, the fossils do show tentative similarities to the Broken Hill skull. We also have Saldanha from South Africa, which has not been properly dated, but has a rather imprecise date of 600,000 years ago, which has also been linked with the Broken Hill and Dutu skulls morphologically, as well the Bodo skull from Ethiopia, which is even older at 600,000 years ago, shows close resemblances with Broken Hill, um, in particular in the large brow ridges and the large cranial capacity. Now, it's tempting to look at these fossils and consider them as a distinct lineage of robust, large browed humans that had lived throughout Africa and seemingly coexisted with the earliest Homo sapiens ancestors. And indeed, some researchers have proposed that, even going as far as to give them a distinct name, like Homo rhodesiensis,
Mm.
or more recently, Homo bodoensis. Um, Perhaps, alternatively, there are multiple populations represented here. Likewise, others have argued that there is a unity in these specimens with those of Western Europe, as we'll see on the next slide. So European remains from Kokabas in Turkey, dating to sometime between 1.5 to 1 million years ago, have in the past been linked with Homo erectus. But a recent analysis by Tommaso More and colleagues from 2024 shows that this hominin lies outside the variation of that species. Thus, we enter into the questionable fossil material of Western Eurasia. At one end of this range, we have the Narmada remains from several localities along the Narmada River in northwest India. The best remains include a partial cranium, limb bones, and parts of the chest region from several individuals that have been variously dated across a wide swath of time, between 300,000 to as late as 40,000 years ago. This, along with the variability in remains, has led to many interpretations of this material, with a recent 2020 paper by Anek R. Shankayan arguing that the remains represent two distinct populations altogether: an earlier robust hominin akin to the aforementioned African material, and a later short and stocky hominin, which they had named Homo normadensis, although that is not a name that has a consensus at the moment. In Southwest Asia, we have the curious phenomenon of the Nesher Ramla hominin from present day Israel. They date to a rather late period of between 140 and 120,000 years ago. Along with the Narmada people in India, these remains would represent some of the last non sapiens or non Neanderthal hominins in Western Eurasia, and their anatomy is very reminiscent of both ourselves and of the Neanderthal, which has led to a hypothesis that perhaps they represent an early diverging population, or a deme, that split off from our common ancestor with Neanderthals and had inhabited Southwest Asia as a contemporary group of hominids. While one contesting view is that these are nothing more than some form of basal Neanderthal. So moving into Europe, we find specimens like Tottevel, aka Arago from France, Petrolona from Greece, and Soprano from Italy, hominins with features very clearly distinct from the earliest species like Homo erectus or Homo antecessor that have raised much discussion. Anatomical similarities with African fossils like Bodo and Broken Hill have led to the suggestion about whether they should be classified with each other as a single species, and usually that name is designated as Homo heidelbergensis, which has a very contentious history. In short, that name was originally coined for a mandible with teeth that was discovered and described in Heidelberg in Germany in 1907 by Otto Schoenstock and Daniel Hartmann. The jaw, which is dated to an impressive 609,000 years old, thus became the type specimen from which all the subsequent Middle Pleistocene fossils could be related to. Now, the obvious problem with this is that when you try to compare crania to a mandible, you're going to run into uncertainties. And for many years, a wide swath of fossils from across Afro-Eurasia were subsumed into this species, with a widely accepted model that it represented a large and vast ancestral population from which Homo sapiens evolved in Africa and Neanderthals evolved in Western Eurasia. But with the hindsight of recent years, it has become clear that much of this fossil material could not all reasonably be classified as one species, and discrepancies in the dating of speciations and fossil localities meant that the common ancestor model would fall flat as well. So increasingly, the name Homo heidelbergensis is being shown the door, or at least reduced significantly to only a handful of fossils. Amidst much new research, two papers stand out to me. The first is a 2024 paper by Walter Neves and colleagues, which analyzed 86 hominin skulls from across the Pleistocene epoch using morphometrics. They found that, taken together, the various aforementioned hominins that are found in Africa, Europe, and will be discussed soon in Eastern Eurasia, do not fall together within a single plot. Instead, they each group separately in a midway point between the Homo sapiens and Neanderthal samples, which would then necessitate that they be classified as distinct taxa. And the second paper is one of the three 2021 analyses of the Chinese Harbin skull, which actually included a phylogeny with a large sample size of Middle Pleistocene material. They found that much of this Middle Pleistocene fossil material formed a successive sequence of clades that eventually split off from the last common ancestor of sapiens, Neanderthals, and Denisovans. Fossils like Bodo, Soprano, Saldanha, Petrolona, and Broken Skull formed one big clade, and Maurer and Tottevel formed another clade, and others like Narmada formed their own clade. Thus, it seems that the ancestral roots of our common ancestor with other hominins, like Neanderthals, consisted of a highly diverse range of forms that must have diverged and perhaps reconverged through interbreeding across many hundreds of thousands of years, and that the possibility of multiple origins for the members of the muddle in the middle should be considered. Of course, ancient DNA would be a godsend. At the very least, we now have a growing number of studies using morphometrics and other methods that are making sense of this material finally. That they all have something to do with each other is suspect. But lumping them all as, say, Homo heidelbergensis or any other name is considered premature at this time. So let's move to the next slide from here. And it's at this point now that we can consider the members of that last common ancestral lineage.
Hmm.
There is somewhat of a bit of disconnect at the moment. Most ancient DNA studies support that Neanderthals and Denisovans consist of one branch, while Homo sapiens consists of the other. While some recent morphological work, namely the Harbin papers, had then pointed to Denisovans and other members of the East Eurasia Middle Pleistocene to be our sister species to the exclusion of Neanderthals. Whatever the case, we can be confident that all these hominins are close relatives of each other, and that they were more than capable of interbreeding, and indeed have done so many times in the past. But we'll save discussions of hominin admixture for the next episode. But for now, I want to make note of two things. One is that interbreeding between Denisovans and sapiens has been documented in the ancient DNA record more so than between Neanderthals and sapiens. And two, despite this record of interbreeding, recent reviews of material by researchers like Andrea Meningazin and Massimo Bernardi have argued that the clear morphological distinctions of hominins like the Neanderthals compared to Homo sapiens makes the case that they were well on their way to being distinct species even as admixture was occurring. As such, there is good reason to retain the distinction of these taxa as their own species, rather than, say, calling the Neanderthals Homo sapiens neanderthalensis,
Hmm.
making them subspecies. The Middle Pleistocene fossil record of East Eurasia has come into much attention in the last decade or so, mainly because of the exciting discovery of the Denisovans in 2010 from the extracted ancient DNA of a diminutive finger bone on the Denisova cave in Siberia, shown here on this slide. That analysis revealed an entirely new lineage of hominin distinct from ourselves and from Neanderthals, but clearly closely related to us, which lived, according to the most recent dates, between 217,000 and 51,600 years ago. Because these original fossils were so fragmentary, paleoanthropologists have eyed earlier discoveries from this region and across Asia to see if their fossils had already been discovered. And this is helped from research using DNA methylation, which compares the regulatory switches of specific genes to those with known analogs to infer physical traits. Essentially, you're taking the DNA and you're making statistical probabilities for what the fossils would have looked like, what the anatomy would have looked like, which is a neat process. These show that Denisovans were very much like Neanderthals in having elongated faces and stocky bodies, though their skulls were somewhat more robust, with massive flat cheek teeth. Further discoveries on newer fossils that have been matched with Denisovan DNA seem to support this, and show that Denisovans had evolved into a number of large subpopulations across eastern Eurasia and perhaps into Oceania as well. Even prior to the discovery of the Denisovans, the fossil record of China has provided a large number of fascinating specimens that date to a period of between 350 and 150,000 years, and show ancestral characters quite distinct from living humans, as well as Homo erectus, but more like Neanderthals. For example, there is the Dali skull from Jiangxi and the Jinshuan skull from Liaoning, both dated to 260,000 years ago, the Hualongdong skull from Anhui, dated to 300,000 years ago, the Maba skull from Guangdong, dated to 215,000 years ago, and the recently described Harbin skull, dated at least 146,000 years ago, among others. Researchers have noticed clear morphological patterns in these fossils. For example, they have massive skulls, with a cranial capacity ranging from 1120 to as much as 1420 cubic centimeters, which is within the range of both Neanderthals and sapiens. Phylogenetic work allies these remains together, and tentatively with material genetically linked to the Denisovans. So some paleoanthropologists suspect that with these, we may have finally given the Denisovans a proper face. But wait, say others. Recent papers by Zhuji Wu, Christopher Bay, and colleagues have taken notice of specimens like the Zhu Shang and Yu Jiao skulls, which date to between 220 and 100,000 years ago. In some cases, their cranial capacity is larger, reaching 1,800 cubic centimeters. That's the largest of any known hominid. And they had wider skulls than the other material. So these forms have thus been given a new name, the Jularens, or the big-headed one, Homo jularensis. But current analyses have shown several conflicting possibilities.
Hmm.
So they may be related to Denisovans, but on a separate lineage from them, or they may be a population or dene within the Denisovan lineage, or they may be the result of admixture between different hominins from East Asia and beyond. It's very unclear. This is all very new stuff, and the phylogenetic studies haven't really helped clarify things either. What we can say for certain is that Eastern Eurasia was a hub of hominin evolution that gave rise to and supported diverse populations, some of which contributed to our own evolution as our species expanded into Eurasia. So let's move to the next slide from here. And at last, we arrive at the most familiar ancient humans of all, Homo neanderthalensis, the Neanderthal. Ancient DNA research indicates that the earliest known fossils of ancestral or plesiomorphic Neanderthals were around as early as 430,000 years ago, represented by the wonderful cache of fossils from Cima de los Huesos in Spain. So-called classic or apomorphic Neanderthals, having their characteristic elongated skulls tipped with an occipital bun on one end and projecting jaws on the other, are found in the fossil record starting 130,000 years ago, with the last ones definitively extinct by 39,000 years ago. Even though the species seems to have come into its own in Europe, by the time the classic morphology evolved, Neanderthals could be found as far as Siberia, Central Asia, and the Levant, with the possibility of more eastern expansions having been discussed here and in the past. There is an apparent contrast in skeletal differences between these regions, and evidence from the ancient genomes of many different Neanderthals point to several large subpopulations having evolved over time. As the Denisovans had done, so Neanderthals seem to have come into contact with us more than once, with evidence that early members of both lineages came into contact between 316 and 219,000 years ago. Neanderthals were robust hominins. The reconstructed skeleton on the far right should give some clue to that. Thick vertebrae, a wide flaring rib cage, and stocky limbs. Though we know that they lived in diverse habitats, there is reason to speculate that Neanderthals were originally adapted to regions with a cool and dry climate. Models point to Neanderthals with a higher metabolism than Homo sapiens and needing a higher caloric intake, which has informed models of their dietary needs. Though they are popularly thought of as primarily carnivorous, as supported by the abundant evidence we have of their hunting of megafauna, we do know now that Neanderthals ate as diverse a diet as Homo sapiens foragers had done. They just needed more of it, especially to support their brains, which were often larger in cranial capacity than our own, large reaching 1750 cubic centimeters. The implications of these nutritional demands will be explored in a later episode, but overall, beyond these key traits, there was much about Neanderthal physiology that was similar or identical with ourselves. In fact, if we move to the next slide, we will finally meet ourselves. We finally reach the end of our journey through the tree of life, humans of our own species. The earliest Homo sapiens fossils are the Jebel Earhout individuals, dated to between 315 and 286,000 years ago. At first, this material was thought to be so dissimilar to living humans today that it was classified as a North African Neanderthal. But recent work beginning in 2017 by Jean-Jacques Hublin and colleagues has offered a reappraisal. Though the skull has thick brow ridges, an elongated cranium, and relatively large teeth, more features of the skull match modern humans in having a flattened face that has sunk and so expanded the forehead instead of protruding high in the face. The presence of a so-called inverted T chin instead of a lack of a chin altogether, so on. Though far from complete, a close morphological match to the Jebel Earhout skull is the Florisbad individual on the opposite side of the continent in South Africa, dated to 259,000 years ago. In Ethiopia, East Africa, the Omo and Hirto remains date to 233,000 years ago and 160 to 155,000 years ago, respectively. These fossils, in contrast, have the rounded globular skulls very close to living humans, and not the ovular skulls of some other early Homo sapiens, like the Jebel Earhout, though they still retained thick brow ridges among other ancestral traits. Just for reference, other East African fossils include Elei Springs from Kenya, dated anywhere between 300 and 200,000 years ago, Ngoloba from Tanzania, dated to about the same time, and Guamde from Kenya, dated at 300 to 270,000 years ago. Similar fossils and other traces are known from Greece and the Levant at this time as well. In prehistory, Africa, in a sense, included much of Southwest Asia. Um, we'll get to that in a later episode. So, right away, what we find is that early Homo sapiens was already widespread across much of Africa and Southwest Asia between 300 and 200,000 years ago. And that while many sport key traits associated with living human populations, there is a clear variation between them all. This has prompted paleoanthropologists like Chris Stringer and Eleanor Sherry to propose that rather than evolving in one region in Africa and then spreading elsewhere, Homo sapiens developed from several source populations across the continent. At least two, but perhaps more, that has diverged and then reemerged multiple times until the whole suite of morphological traits came together to give us the modern human. This African multi-regionalism, or mosaic evolution, has been detected somewhat in recent genetic studies, where present-day African hunter-gatherer groups have very deep ancestries indicative of ancient population splits. And morphometric studies on human cranial and mandibular remains in Africa have found continuity between the early fossils and more recent populations within the last several thousand years. That's not to say that every ancient fossil would necessarily have been a direct ancestor to living people today, but in the absence of ancient DNA from these remains, we just can't be sure which remains come from populations that died out or not. In all, while there are always exceptions within the entire global population, for example, some living peoples will have prominent brow ridges or occipital buns or robust skeletons, and these may be expressions of ancient gene flow. In general, Homo sapiens can be defined morphologically as follows: having a rounded globulous skull, a flat sunken face lacking significant prognathism but providing a high forehead, a pointed inverted T-shaped chin, a derived dental morphology in both size and crown shape, and postcranially, a cylindrical rib cage and a less flared pelvis. Though the phrase anatomically modern human has fallen out of favor due to this new understanding of mosaic evolution, Homo sapiens today, for all its physical variation, remains one very closely related species genetically. Um, but with that, we reach the end of our survey of the hominin fossil record. Albert, do you have any closing thoughts you'd like to give us?
No, that was a really great overview of who's who in the hominin family tree. It's really amazing just how much material we actually have. I mean, it's almost a cliche to point this out when discussing human evolution, but it's the classic question of where are all the missing links. Well, it turns out that we have more than we know what to do with in some cases. It has certainly been an excellent explainer of what we currently know regarding the fossil record of our lineage.
Oh, I'm glad to hear that. And definitely, this is a welcome update to the previous season one as coverage of hominin evolution, even though it was much shorter. But obviously, a lot of this material will be stretched out in the accompanying episodes. But alas, we've reached the end. So let's move to the next slide and see what we have next in Story of Humanity Prologue. So the next episode, we're going to dive deeper into this apparent contradiction between physiology and genetics. So yeah, we're going to tackle the topic of human diversity, commonly associated with the concept of race. We're going to see how the idea of race developed, what it meant then, and what it means now, and we'll explore how biological anthropologists currently understand human diversity from a huge volume of research available to us, including the incredible sweep of human migrations, population expansions, and admixture events that brought our species to nearly every region on the Earth, using only feet and brains. So that should sure be a fascinating episode that I very much look forward to checking in.
Mm-hmm.
But moving on to our general acknowledgments, of course, we are on Patreon. It's patreon.com/timingclades. Patreon is a website where you can go in and receive updates from content creators, often ahead of public releases. You can provide monetary donations to support their projects, usually through a series of tier lists where you can get different perks. And of course, these contributions help us continue the series and develop new projects and expansions. So, for example, on one of our upper tiers, you can get a shout out for our episodes. So, of course, we want to thank my sister Julie and our good friends Denver, Frankish, and Valdemuzio for their support for the series. It's always appreciated. Moving on from here, of course, we want to thank our good friends Emily and Alicia for their contributions to this series. Emily is responsible for the wonderful theme music that opens up each episode, and Alicia is responsible for the color scheme of Albert's Alvarezsaur avatar. Cheery little fellow. Moving on, yes, we are still on Twitter at TimingClades, where usually we post episodes that when they premiere. Though most likely you are watching us on our YouTube page through Time and Plate, so if you wish to like and subscribe, always more than welcome to do so. If you have any questions at all about this series, whether it be about the hominins and topics discovered here, or just anything, there's three ways to contact us. You can email us, timeandclates@gmail.com. You can tweet at us, or you can comment on our YouTube page, and we will respond in kind. And as well, if you want to see the description on our YouTube page where we have all of our references, that's a very great idea. There will be a long Google Doc provided showing all of the papers referenced for this episode, so you can follow these discoveries for yourself and read the nitty gritty details on your own. But with that, that is the end of the series. Of course, we have much more through time and clades on the way. We have recorded our new lecture series by our friend Alari on paleoart methods that's currently in progress, as well as future installments of Humanity a Prologue to look forward to, and who knows what else. So as always, we thank you all for your support and for joining in with us on this journey today, and we wish you all a very pleasant afternoon.
Indeed. Take care, everybody.